Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 2. This is lecture number 38. The topic is genetic variation. Discussing agenda for the topic is genetic variation. Lecture outcome. After watching and listening this lecture, students will know about various mechanisms through which the genetic variation occurs. Introduction. Genetic variation makes evolution possible. In the origin of species, it was the famous manuscript published by Charles Darwin in 1859. In that book, he documented the theory of theory of evolution by means of natural selection. In that book, Darwin provided abundant evidences that life on Earth has evolved over time, and he proposed natural selection is the primary mechanism for that change. He proposed that individuals differ in their inherited traits and that selection acts on such differences, leading to evolutionary change. So Darwin was of the opinion that the species tend to change with the passage of time. They thrive to adopt themselves to the prevailing environmental conditions, and if they have such traits which enables them to become more adapted to the given environmental conditions, then such type of traits become more common in the population, and such individuals are at a greater chance of reproduction and leaving more offsprings, and such favorable traits are propagated in the population, and they are selected by the nature. Although Darwin realized that variation in heritable traits is a prerequisite for evolution, he did not know precisely how organisms pass heritable traits to their offsprings. At the time when Darwin was documenting his theory, there was no knowledge of genes or alleles or DNA or heritable material or things like that, but there was only one theory about inheritance, which was blending inheritance, and the blended inheritance said that both the parents share their characteristics in equal proportion to their offsprings. So Darwin recognized the fact that variations should be passed to the offsprings, and these variations should be favored by nature, but he didn't had any idea and didn't had any knowledge about the mechanism through which those traits are passed. Now we know that those traits are passed through genes, through alleles, but he didn't have any idea at that time. Just a few years after Darwin published The Origin of Species, Gregor Mendel wrote a groundbreaking paper on inheritance in pea plants in 1867, means 8 years after the publishing of this document. Mendel published his work, and Mendel introduced the particulate inheritance theory. He proposed a model of inheritance in which organisms transmit discrete heritable units, now called genes, to their offsprings. Although Darwin did not know about genes, Mendel's paper set the stage for understanding the genetic difference on which evolution is based. It is an interesting thing to share here is that after the death of Darwin, that manuscript of Mendel was also found in his library or repository or documents, but for some reason he was unable to study it and modify his theory of evolution or update his theory of evolution according to the Mendel's findings. Here we will examine such genetic differences and how they are produced. Individuals within all species vary in their phenotypic traits. Just take an example of the human population. There are so many variations in the hair color, skin color, in the height. Although we belong to the same species, but we do have differences in the phenotypic traits. Among humans, for example, you can easily observe phenotypic traits, phenotypic variations in facial features, height, and voice. Indeed, individual variation occurs in all species. And though you cannot identify a person's blood group from his or her appearance, this and many other molecular traits also vary extensively among individuals. Within our bodies, the nucleotide sequences, they also vary considerably from each other. Such phenotypic variations often reflect genetic variations. I hope you already know that the phenotype is controlled by an underlying genotype, although sometimes the genes are under the effect of the environment. There is a phenomena called epigenesis involved, but predominantly whatever is there in the genes will be expressed in the phenotype. So the phenotypic variations are because of the differences in the genes, the differences in the genetic makeup, differences among individuals in the composition of their genes or other DNA sequences. Some heritable phenotypic differences occur on an either or basis, such as the flower colors of Mendel's pea plant. Here what they want to say is that in some of the phenotypic traits there are two extremes, means they are referring to the typical Mendelian crosses where there were no intermediates, either the seed was round or wrinkled, or the plant was tall or dwarf, or the flower was of one particular color, or the seed color was yellow or green. There was no intermediates. Characters that vary in this way are typically determined by a single gene locus with different alleles producing distinct phenotypes. In contrast, other phenotypic differences vary in gradation along a continuum. For example, height. Now there are no extremes in the human height. If you go out to a market, you will find people with varying degrees of heights. So it is a continuum. Such variations usually results from the influence of two or more genes on a single phenotypic character. So such traits are usually polygenic, means they are controlled by more than one genes. In fact, many phenotypic characters are influenced by multiple genes, including coat color in horses, seed number in maize, and height in humans. How much do genes and other DNA sequences vary from one individual to another? Means we are different from each other, and these differences are because our genetic material or DNA sequences are different from each other. So how much these DNA sequences are different from each other? Genetic variation at the whole gene level can be quantified as the percentage of the loci that are heterozygous. Means what is the percentage of the various gene loci that are homozygous for a particular allele or heterozygous for a particular allele. As an example, on average, the fruit fly Drosophila melanogaster is heterozygous for about 1920 of its 13,700 loci. So it means there are 13,700 loci where the alleles exist, and out of them there are 1920 loci which are heterozygous, means where dissimilar copies exist, and in the rest of them similar copies or homozygous copies are present. So what makes this percentage? 14% of the loci in Drosophila melanogaster are heterozygous. Considerable genetic variation can also be measured at the molecular level, which is nucleotide variability, but little of this variation results in phenotypic variation. Why? This is the answer. Many nucleotide variations occur within introns. Introns are the non-coding regions. These are removed during the process of splicing. So variations or differences amongst individuals in those areas will not be expressed in the phenotypes because the differences in those areas will not have any effect on the proteins. Here you can see the phenotypic differences within our population. The horses belonging to the same population, the same species, yet they show phenotypic differences in their coat color. And just mentioned earlier that such phenotypic differences with such varying degrees, if a trait show such variable degree of differences, then it is an indication of the fact that this trait is controlled by multiple genes. On the other hand, if a trait is just two or very few variations, then it is usually controlled by a single gene with one or two alleles, different alleles. And of the variations that occur within exons, exons are the expressed regions, the coding areas which codes for the proteins. Most do not cause a change in the amino acid sequence of the proteins encoded by the gene. And what could be the answer for that? That exons are the coding areas, and the differences occurs in the exons, but still there are no differences in the protein encoded by the gene. So students, what could be the answer to that question? Press on your mind. For example, in the sequence comparison shown in figure 2, there are 43 nucleotide sites with variable base pairs where substitution have occurred, as well as several sites where insertion or deletion have occurred. Although 18 variable sites occur within the 4 exons of the ADH gene, only one of these variations at site 1490 result in an amino acid change. So there are variations, there are changes in the nucleotide sequences at various points here, but these variations are not resulting in a change. So there are 18 variable sites and they occur in the 4 exons, and only one of these variations at site 1490, means somewhere here, results in an amino acid change. So why not these other changes are resulting in the amino acid change? Maybe the answer is the redundancy of the genetic code. Note, however, that this single variable site is enough to cause genetic variation at the level of the gene, and hence two different forms of the ADH enzyme are produced. So the information encoded by the gene is so much sensitive that sometime a change of a single amino acid results in the change of the protein, because these proteins, after they are translated, they mold into specific three-dimensional structures, and during that conformation into 3D structures, each and every amino acid, they have their own role. So when one amino acid is replaced by another, then the structure is slightly changed, and for proteins, the structure determining the function principle is extremely important. So the function is changed to a slight degree in that case. It is important to bear in mind that some phenotypic variations does not result from genetic differences among individuals. Here, figure 3 shows a striking example in a caterpillar of the southwestern US. Phenotype is the product of an inherited genotype, and many environmental influences already discussed that sometimes the phenotype is also under the effect of the environment. In a human body, bodybuilders alter their phenotype dramatically, but do not pass their huge muscles on to the next generation. In general, only the genetically determined part of phenotypic variation can have evolutionary consequences. So if variation occurs at the genetic level and this variation is favored by nature, then only that variation will be passed on to future generations and will be retained in future generations. As such, genetic variations provide the raw material for evolutionary change. Without genetic variations, evolution cannot occur.